All right, I gave you 5 minutes to draw step by step the phases of mitosis. Yes, this is the same sheet that you did last week. Yes, I do expect you to be able to draw this on your test on either Monday, November 21st, or Tuesday, December 21st, or Tuesday, December 22nd, the days before break. I will ask you on your test, step by step, to either draw mitosis or to give me all the, using words, the key events of mitosis. Most people tend to do the drawing option. So what I'm going to do, since we've already drawn this multiple times in class, I am going to use a copy that we already drew and go through this fairly quickly. Okay, this is all referenced in our videos from in-person agenda if you were gone. These are all referenced on our in-person agendas as images on the bottom of the slides in Canvas. So I'm not going to spend tons of time with this because we've already talked about mitosis many times. During interphase, 96% of the cell's life, there's a nucleus, there's genetic information in the form of chromatin that's uncoiled within the nucleus. During G1, the cell grows. During S phase, the DNA replicates. During G2, it prepares for replication. He says it does its day-to-day function. Now, if you got something wrong, if one of your pictures is wrong, I'm not asking you to write out the steps again. You already have this sheet. But if one of your steps is drawn incorrectly, I would suggest you put a big X through your picture and then draw the corrected version, the corrected picture in the right-hand column, because yours looks slightly different than this. Remember, puppy mitosis needs a PMAT, where the P stands for prophase. Prophase. So during prophase, the nucleus dissolves. We have, oops, wrong one. The nucleus dissolves. We have chromosomes. We drew 4 chromosomes. We get these things called spindle fibers starting to show up. We have chromosomes, big X-shaped condensed DNA. Remember, these are sister chromatids that are genetically identical to one another. This came from mom, this came from dad. This one came from mom, this one came to dad. These small chromosomes are homologous chromosomes. These big chromosomes are called homologous chromosomes. Our puppy mitosis needs a PMAT. We're on the that stands for metaphase, sorry. That stands for metaphase. The other M word that goes along with metaphase is middle. Meta, middle, middle, meta, meta, middle, middle, meta. Remote until 15. So, but she won't be here tomorrow. Meta, middle, middle, meta, meta, middle, middle, meta, meta, middle. The chromosomes go to the middle. Now, the way that this is drawn is red, red, green, green is random. It doesn't matter if you don't have them in that order. But the chromosomes line up in the middle. We have 4 chromosomes. We need 4 chromosomes lined up in the middle. The order is random. It doesn't matter the order in which these are put here. This could go red, green, red, green. Oh, I'm sorry, red, green, green, red. Okay, doesn't matter. It's random. It's random. Meta, middle. Why do they go to the middle? The chromosomes go to the middle because of the spindle fibers. Pull them to the middle. Meta, middle, meta, middle, middle, meta. Ana, apart, or away. The sister chromatids pull apart. The sister chromatids are pulled away from each other. Ana, apart. Ana, apart, apart, ana. Why? Because of spindle fibers. The spindle fibers are pulling those things apart or away. Ana, apart. PMAT, we're going to slash telophase into cytokinesis. We get this pinching of the cell membrane right here. The nucleus starts to reform. And then what's going to happen is our chromosomes is going to go back to chromatin. It'll uncoil. But what we have is we have 1, 2, 3, 4, 1, 2, 3, 4. This sister chromatid is the exact same as this. This one's the exact same as this. This is the same as this. This is the same as this. We have exact same sister chromatids because the sister chromatids are genetically identical to one another. And the final product is we're going to end up with we're back to interphase again, 2 genetically identical diploid daughter cells. We start with 1 diploid cell, we call it a parent cell. We end up with 2 genetically identical diploid daughter cells. It has a complete set of genetic information. We started with 4 chromosomes in prophase. We have 4 chromosomes in telophase. Each cell has 4. They're genetically identical. They have a complete set of DNA. They got 1 copy from mom, they have 1 copy from dad. That's mitosis. Questions about mitosis? All right, today we're going to draw meiosis. Meiosis has some similarities to mitosis. It has some similarities, meta, middle, ana, apart, but there are definitely some differences too. So, on that desk is a sheet very similar to mitosis, except for now we're talking meiosis. I'm going to use the same colors that I did last week. I'm going to use a red and green colored pencil. If you don't have a red and green colored pencil, I would encourage you to grab a red and green colored pencil. Mitosis, what you just drew, makes body cells. Body cells are diploids. They have a complete set of genetic information. Half came from mom, half came from dad. Meiosis is the production of what we call sex cells, sperm cells, or egg cells, depending whether we're talking male or female. There are some similarities. It's still PMAT, prophase, metaphase, anaphase, telophase, or if you don't like PMAT, pass me a taco. It just happens twice. So there's Meiosis I and Meiosis II. PMAT happens twice. Pass me a taco, happens twice. Okay? Now we tried to kind of bullet point the kind of key phases or the key processes here, so we didn't have to write as much. But during G1, the cell is going to grow, or we would say it increases in size. During S phase, we duplicate the DNA, and then in G2, the organelles are going to be replicated. Okay, that's the big things. Interphase is pretty darn similar, and we start with 1 giant cell. The giant cell has a nucleus. We're going to label that. And it has genetic information as a uncoiled bowl of spaghetti that we call chromatin. From a tin uncoiled. Okay, it's uncoiled DNA. Now, I should probably take a step back. I want to talk big picture. 1 diploid cell, through the process of meiosis, is going to create 4 genetically different haploid sex cells. One diploid cell goes through meiosis, and at the end we end up with 4 genetically different haploid sex cells. That's the big picture thing here. We start with a diploid, we end up with 4 genetically different haploid cells. Now right here we can't really, it doesn't really look like a diploid. I'll show you the diploid in a moment. So some of these lengths are too small, like for the example in Prophase One. So PMAT happens, but each chromosome, that's kind of really small to write the word chromosome in there, but each chromosome pairs with its corresponding homologous chromosome and forms what is referred to as a tetrad. At this point, portions of the chromosome swap, and this causes what they call crossing over, which allows for genetic variation. Crossing over. Crossing over was a slide I put up last week. During Prophase One, I like to consider Prophase One as like buddy check. You guys ever been to a summer camp? You go swimming, you have a buddy, you do a buddy check. The intent is as long as you and your buddy, as long as you know where your buddy is, they aren't probably on the bottom of the lake. Buddy check, right? The idea is you grab your buddy's hand and be like, hey, we're together, we're right here. That's what's going on. Homologous chromosomes are doing a buddy check. And tetra, that prefix means 4. So how many? We have 4 chromatids. 1, 2, 3, 4. We have 4 chromatids. These chromatids go through something called crossing over. Because they are so close to each other, right here, the 2 that are really, really close, it can happen, doesn't have to happen. We get this crossing over where basically they cut the chromatids at the same spot and then they swap. Okay, and I showed you a slide of that. That's what we're going to show, and we're going to show that in on all the steps throughout the process. Sorry, video, forgot to show you. Where that black line is, they get cut, and we end up with those 2 chromosomes on the bottom that have some swapped tails. Okay, so that is what we talk about when we talk about crossing over. So Prophase One, our puppy needs a PMAT. What happens? The nucleus has to disappear. Very similar to prophase during mitosis. And we are going to draw buddy check. So we're going to have a big red chromosome. I'm going to leave just a little tiny bit of that bottom off. And then I'm going to have a big green chromosome that's like right there. And I'm going to leave just the bottom part of that off. Now, actually, I'm going to draw it. They're going to be over each other. Okay, right here, that's called crossing over. And what's going to happen is they are going to cut and they're going to swap. Now, I'm going to only show crossing over on 1 pair of chromosomes. It could happen on both. It could happen on the top and the bottom. It could we call that a homologous chromosome, or a homologous pair of chromosomes. Okay, this is a homologous pair. This is a homologous pair. We have two homologous pairs. Now we could draw more if we really wanted to, but I'm not going to. Our puppy needs a PMAT, so we go, oh, we need spindle fibers. Sorry, forgot spindle fibers. Spindle fibers. Our puppy needs a PMAT, so M for meta, M for middle. Slight difference of what goes to the middle here, though. Now what goes to the middle are my homologous chromosomes. Okay, they line up along the middle. So I'm going to take my big red chromosome, and I'm going to draw it here, and I'm going to draw, but I'm going to show that crossing over occurred. So there was a slight swap of genetic information. So there's a tip of green on that one. My green chromosome, big green chromosome, has the tip that's going to be red. That's the crossing over that occurred. Then I'm going to draw my other homologous chromosomes in the middle. They're in the middle because of spindle fibers. Okay, these homologous chromosomes are in the middle. So we can say that spindle fibers, again, blank's not really big enough. Spindle fibers attach to the chromosomes, and they line up individually and randomly. And this should be in, not on. In the middle of the cell. The reason they're lining up in the middle is because of the spindle fibers. This randomness, this random lining up, randomly how they line up, is called independent assortment. And it allows for further genetic variation. Last week, we spent some time talking about the advantages of genetic variation. On Monday or Tuesday, December 21st or 22nd, you should probably be expecting to see something about what are some benefits of genetic variation. Now, what this randomness is, what we're saying. I drew this big red chromosome here. There's a big green chromosome here. How these two lined up is random. You could have I could have just as easily drawn this as green and this is red. They are random, and they are absolutely have no bearing on how these line up. These lining up this way is independent upon how these lined up. Called independent assortment. Now we have 23 pairs of chromosomes. Half came from your mom, half came from your dad. How many different genetic combinations are there just by independent assortment on how chromosome number 1 lines up, how chromosome number 2 lines up, number 3 lines up? In order to determine it, it's 2, because you have 2 choices, mom or dad or mom, and there's 23 different cells, chromosomes, so it's 2 to the 23rd power. It's in the 8 millions. You have 8 million genetic combinations that you could make with your sex cells by independent assortment alone. That's not including crossing over events occurring. So the likelihood of 2 eggs being genetically identical is very, very low because of independent assortment and crossing over. So meta-middle. It's very similar to mitosis, where things line up in the middle, but the homologous chromosomes go to the middle. The homologous chromosomes go to the middle, not the chromosomes. The pairs go to the middle. Ana still stands for apart, but this time the things that get pulled apart are the things that lined up in the middle from each other. So the homologous chromosomes lined up in the middle. So now the homologous chromosomes are going to get pulled apart. So that big red chromosome is going to get pulled to the right, and this big green chromosome is going to get pulled, I'm sorry, this is getting pulled left, this is going to get pulled to the right. They're going to pull apart, they're going to pull away from each other. Little red, little green. Because of spindle fibers. They're getting pulled apart. They're getting pulled away from each other. The spindle fibers pull the homologous chromosomes towards the opposite sides of the cell, or apart. These are getting pulled apart or away from each other. Meta-middle, ana apart. Homologous chromosomes go to the middle in metaphase 1, and anaphase 1, the homologous chromosomes get pulled apart. Telophase, we're going to slash this like we did before with cytokinesis. During cytokinesis, we get this pinching in of genetic information of the cytoplasm, the cell membrane. The nucleus reforms. So we get the nucleus that starts to reform. And now what we have is these two chromosomes are going to be in this cell. These two are going to be in this. Now we haven't actually gone all the way through, but these will pinch completely into two new cells. So we end up with a big red chromosome that has just a little bit of a green tail on the one end, and a little green chromosome. And I end up with a big green chromosome that has a little bit of red on it, as well as a little red chromosome. And I'm drawing them just in any random spot. It doesn't matter. They're floating around in the cell. The nucleus reforms. And what happens is they separate into two haploid cells. Up in prophase, how many chromosomes did we have in prophase? 4. Good. We had 4. How many do we have now in each cell? 2, which is half the amount of genetic information. Haploids have half the amount of genetic information. How are we doing on that? I want to move on, make sure we're all pretty good. Okay. That's meiosis 1. Very similar to mitosis, except for homologous chromosomes go to the middle. Homologous chromosomes pull apart. After meiosis 1 comes meiosis 2, and meiosis 2 is almost identical to mitosis. Things go to the middle. Sister chromatids pull apart. Same idea, except for now we have half the number of chromosomes that we started with. We have haploid cells that are going through basically mitosis, and they're going to go through and they're going to have half the amount we're going to have half the amount of genetic information than we originally started with. Okay, anything you need to see again before I go to the other side? Okay, so then after PMAT happens in Meiosis I, then PMAT happens again in Meiosis II. In Meiosis II, the cell immediately goes into what is called Prophase II, Roman numerals, without replicating the DNA. Without replicating the DNA. They don't replicate it. It already was replicated. That happened in S phase during interphase before this all happened. It has the same events as prophase from, as mitosis. As mitosis. Now we have two cells now. So I'm going to draw my two cells side by side because that is how we split them on the previous page. The nucleus dissolves right away in prophase II. You could say, wait, you mean in telophase it reforms and then immediately goes into prophase II and it dissolves again? I think so, yeah. I don't know why, but it does. That's what I've read. So during prophase II, we now have, I want to make sure I have my chromosomes the same. I have a big red chromosome over here that has a little tail of green on it, and I have a little green, and then I have a big green that has a tail of red and a little red. And I'm just drawing them in any random spot in the cell because it makes absolutely no difference where they are. PMAT happens, so we go prophase II, and we go metaphase II. Now, we have spindle fibers. Spindle fibers are going to pull these things apart. And these spindle fibers are attached to something called a centriole. That's the first time I've mentioned a centriole. But these are the dots, these are the anchor points, these are the locations where spindle fibers come out of. I'm going to draw my spindle fibers coming out of the North Pole and the South Pole in each cell. We could go east and west. Then they're going to divide a little differently. I think it kind of shows up on your sheet a little nicer this way. So I'm going to go North Pole and South Pole is where I'm going to put my spindle fibers. And the reason I'm going to do that is because when I draw meta-middle, okay, so what happens here is the chromosomes line up in a similar way to the metaphase stage of mitosis. It's still meta-middle. Our memory device still holds true, meta-middle, but now our chromosomes go to the middle. So if we're talking North Pole, South Pole, we're going to line our chromosomes up along the equators. So when I draw this, I have a big red chromosome, and I'm going to draw my big red chromosome such that my green little tail is on the southern hemisphere. That's how I'm going to draw mine. Okay, I'm drawing my chromosomes along the equator. Then I have my big green chromosome, and I'm going to put the red portion in the northern hemisphere. Now, how those line up, how these line up versus like this like this like this, that's random as well. I'm just drawing it that way because that's the way I chose. It's all random. increases genetic variation. Why spindle fibers? Anaphase, we have PMAT, Prophase 2, Metaphase 2, Anaphase 2. And during Anaphase 2, we still only have 2 cells. We still have these north and south pole things at the centrioles. Centrioles are at the north and south pole. But now, the sister chromatids separate and move to opposite ends of the cell. Meiosis II is almost identical to mitosis. So now sister chromatids, my big red chromosome, sister chromatid on this side is going to go to the north. My big red sister chromatid that has the little green tail is going to head towards the south pole. My big green sister chromatid that has a little bit of red tail on that is going north. Those are heading south. Sister chromatids pulled apart. Sister chromatids pulled apart. And they're pulling apart at the equator because of how I drew. Now, we could have drawn it so these are all rotated 90 degrees. Then my centrioles are here, and then my sister chromatids would be pulling this way. Could, it's just not how I chose to draw. We're going to call this telophase 2 / cytokinesis once again. And we're going to scribble out that cytokinesis down there. Telophase 2 has the same events as telophase from mitosis. I'm trying to emphasize that if you haven't caught that already. Now, what's going to happen is this cell is going to pinch at the equators. It's going to pinch at the equators. So when we draw our bubble, we're kind of drawing a snowman kind of shape here, where we have the head of our snowman, and we have our body of the snowman. They just happen to be like the same size. You might criticize technique there, but it is what it is. The nucleus starts to reform. The nucleus reforms. The nucleus starts to reform. The nucleus starts to reform. The DNA technically is going to start to uncoil. I'm not going to show it like that yet. So in this top, we have a big red chromatid. We have a little green chromatid. In the bottom, I have a big red chromatid that has a little tail that's green. And I have a little green chromatid. In the bottom one, I have a big green chromatid, and I have a little red chromatid. In the top, I have a little red chromatid, and I have a big green chromatid that has a little red tail on it. Now, I'm drawing them in all different orientation because it doesn't really matter where they're drawn. I'm trying to show that. I said the DNA technically uncoils into chromatin. That's true. I'm actually not going to show you, I'm not going to draw it like that, how to design But what we end up with is we end up with 4 haploid cells, and we can just call it an N. That's what haploid means. That are genetically different. We have 4 bubbles, final picture, 4 bubbles, 1, 2, that should have been separated, 3, 4 bubbles. Each bubble has a nucleus. I'm going to say this once again. Technically, the DNA is going to uncoil into chromatin form. I'm not going to draw it like that. I want to show you what our final kind of product looked like when we're all done. So I'm just kind of transferring those sister chromatids that we did from the top to kind of match that. So the cell membrane has finished pinching. Now what can you tell me about this cell? Let me draw it just a little bit longer. What can you tell me about the DNA in this cell compared to the DNA in this cell? Are they the same or are they different? Based upon colors and sizes. Is that red chromosome the same as that red chromosome? No. No, right? There's that little green tail, so those are different. What about this big chromosome versus this big chromosome? Well, those are different. What about this big chromosome compared to that big chromosome? Different. What about that one? They're different. They're all genetically different from one another. We end up with 4 genetically different. This is kind of just repeating what I wrote on the back. Haploid. Never mind, that's exactly what, I'm not going to rewrite that, sorry. That's like exactly what I have right here. 4 genetically different haploid sex cells. I was just going to add the word sex cell in there. I don't think that's necessary. This is making sperm cells or egg cells. Sperm or eggs. Let's quick go through the process of mitosis. At the end of mitosis, how many cells do we have? Mitosis, what you drew when you first got to class today. 2. Are they genetically identical or different from when they started?
Identical.
Identical. And are they haploid or diploid cells at the end?
Diploid.
Diploid. And they're used for, and I'm just going to say they're used for body cells. Body cells. Meiosis, the picture we just drew. How many cells did we end up with?
4.
And they are genetically identical or different?
Different.
Are they haploids or diploids? Because they have half the number. They are haploids, and this makes sex cells. And when we talk about sex cells, we're talking sperm or egg cells. Hey, Mr. Ather.
Hi.
How are you?
Good.
Good.
That's quite the get-up.
Thank you.
Looks awesome.
Thank you.
I'll come back. I didn't know if you
Just talking about sex cells.
had. Okay.
Egg cells, sperm cells, making sperm cells, egg cells.
All right.
Now I'm going to lie to you, not lie, I'm going to misinform you. During the process of meiosis, 1 diploid cell creates 4 haploid cells. That's a true statement. In males, 1 diploid cell creates 4 sperm cells. That's a true statement. In females, however, 1 diploid cell does create 4 haploid cells. That is also a true statement. However, 1 diploid cell does not produce 4 egg cells. In human females, let me clarify that, in human females, 1 diploid cell does create 4 haploid cells, but of those 4 haploid cells that are produced, only 1 of those haploid cells is what they would refer to as a viable egg. Only 1 of those eggs is capable of being fertilized. Okay? In males, all 4 of those sperm cells are viable sperm. All 4 of those sperm cells that are produced during meiosis are capable of fertilization. The rates of meiosis vary greatly in males versus females in humans. Males have on hand millions of mature sperm that are ready for fertilization. Meiosis occurred a lot at any given point. During sex, millions of sperm are released to try to fertilize that one egg. But in females, there is only one egg produced every 28 days. Okay, so meiosis occurs once every 28 days in females, and humans produce one viable egg, unless it happens in both ovaries. Therefore, we could have like fraternal twins. Sometimes it happens. But in males, meiosis is occurring very rapidly all the time and producing millions of sperm at any given moment. Okay, so the rates of meiosis are very different. Okay, but the idea is still one diploid creates 4 haploid. Just in females, those 4 haploid cells, the 3 are what they call polar bodies. They can't fertilize. They're not capable of fertilization. All right, let's see. We have moments. We're doing, okay, 6 minutes. You have that, you have that. 2 things for homework tomorrow. One thing is there is a video posted in Canvas that you are to watch, and there are 10 Google form questions for you to answer at the end of the video. Take a screenshot of your Google form score. The score is going to reflect what your answer. I'll get your color pencil. The Google form is going to reflect your thing. So if you get them all right, you're going to get an A. If you got them all wrong, though, then you're not going to get an A. You're going to get an NE. Okay? While I'm talking, I'll come around and take your colored pencils. Okay? So that is one assignment, is watch a video, 10 Google form questions that go along with that. Your other homework for tomorrow is on a desk right there or right there. And it is a Venn diagram of, mitosis versus meiosis. On page 2, and it's photocopied the way it is so you can rip it off. On page 2, there are statements that we want you to use. Each one is used once and only once. Compare and contrast Venn diagram, mitosis and meiosis. Take a picture, submit that, or there is a PDF that you could mark up, and you can do it that way as well if you want. 2 assignments for your virtual day homework for tomorrow. Test Monday, December 21st, or Tuesday, December 22nd. If you have any questions, please let me know.